Okay. So Joseph, do you see where it says lesson info? Can you click on that, please? Great. All right, so it drops down and you should see two options there. One of them says a student exploration sheet and another says the vocabulary sheet. For the most part, we won't be working with the vocabulary sheet. We'll just be using the student exploration sheet. And I may refer to this as worksheet. So I'll try to use the same language, student exploration sheet, but it might just be worksheet sometimes. So if you click on Google Doc there, Joseph, yeah, it'll take you to this screen. You'll click make a copy.
Mr. Ed,
Yes, ma'am.
is it possible you could send me a copy of this meeting because I can't hear anything anyone's saying and my screen keeps glitching in and out?
Okay, here's what I would try to, I can, yes, but here's what I try to do first. If you put your mouse at the bottom of the screen, do you see where it says turn on captions?
Oh, yes.
Click that and see if it does it come up as what I'm saying?
No, I don't see anything.
So there aren't any captions that are coming up?
Not on my screen. It's probably because it's glitching again.
Okay. Well, I will be sure to send you a copy of this meeting. Okay, Joseph. So this is what will come up. Now what I want you to do, Joseph, do you see where it says copy of building the title of the document in the top left? Yep. So click on that and change it so that you put your name in it. Delete the copy of, just put Joseph Frazier. Excellent. And then where it says My Drive, click on My Drive. Yep. You're going to want to put it in your biology folder. Yep. Excellent. Okay, so this is exactly how you would walk through doing the assignment. Now that Joseph has made his own copy of the student exploration sheet, he can type into it and all of the directions to actually work the gizmo are right there in the worksheet. So for example, scroll down just a little bit, Joseph. Okay. Let me see. I'm trying to look for a specific part of the worksheet. Oh, sorry. I didn't mean for that to go off again. Are you still sharing, Joseph? Okay, go back to the gizmo, actually, Joseph. Yeah. Okay, so the directions are kind of here and in the worksheet. So if you see that, drag one of those nucleosides on the right side. Yeah, drag it into the, good. And then it says also, so it says on the left, it says drag and join nucleosides and phosphates to build the left side of a DNA molecule. So now that you've dragged the nucleoside in, what you'll want to do is take one of those blue phosphates. Do you see the blue phosphates? Yeah. Drag that in. Yep, good. And now get another nucleoside and attach it as well. Excellent. So It's going to walk you through how you do these simulations, and you're going to answer questions on the worksheet as you go. Okay, so this is how the gizmos work. Thank you so much, Joseph. You can stop sharing your screen now. All right, so we've got 29 minutes, almost 30 minutes left. I think that'll be enough time to get through the lesson for the day. Please let me know if you have any questions on how to access the gizmos. I did also put an assignment in our modules, which is where you'll submit the worksheet once you're done with it. And the directions are there as well. Okay. So today is October 7th, 2020, and we are in the second day of our 5th unit. The title of today's lesson is DNA Replication. DNA Replication. So you should definitely try to write down the title of the lesson and the dates and Unit 5, Day 2. The objectives: biologists will be able to explain the process of DNA replication, which copies DNA during synthesis of interphase. So we should remember that from a couple of units ago when we talked about the cell cycle in Unit 3. So you all know that DNA is replicated, or it is copied during synthesis of interphase. So interphase is 3 parts. We've got G1, S, and G2. During the S phase is when the DNA is replicated. And the essential question that we seek to answer: why is DNA replication an essential process for living organisms? Okay. So first question, why does our DNA need to be replicated? Does anybody have an answer? Again, I want you to think back to our 3rd unit. Why do we need to replicate DNA in the first place? Any thoughts on that? Why do we need to replicate our DNA?
To grow.
Yeah, that's a big part of it. So to grow. So when we talked about, yeah, and Gabby, so we can reproduce. Excuse me. So when we talked in Unit 3 about the cell cycle, we talked about the fact that we produce new cells for several reasons: to replace damaged cells, to reproduce, as Gabby said, and to grow, as Alicia said. And those are all valid reasons. And so for those same reasons, we need to replicate our DNA, because before the cell divides, we have to make a copy of the DNA for the new cells. So you should write that down. Again, what you see highlighted in this lesson should be written down somewhere in your notes. So before the cell divides during M phase, so mitosis or meiosis, there must be a copy of the DNA for the new cells. And this is why we need to replicate our DNA. Okay. So this is what DNA replication looks like. And for those of you who are thinking about taking the AP biology class, we're going to break down all of these different structures that you're looking at. It's a very complicated process, but once you really break it down, it's pretty easy to remember. But what we should take away from this slide is that we've got old DNA over here. This is the DNA from the original parent cell. And throughout this process, what's happening is that we're creating 2 new strands of DNA. So from a single strand of old DNA, we end up creating 2 new strands of DNA. Now, there's a lot going on here as well, and each one of these things serves a vital function in this process. So there's a lot of opportunity for things to go wrong, but our bodies, our cells are so good at doing this process that rarely is there a mistake. Now, I want you to notice another thing on this slide. We've got an ace there, an ace there, ace there, ace there, ace there, and ace there. What do all of these ASE endings mean? Does anybody remember what ASE means?
Are they like types of enzymes?
Exactly, exactly. So anytime we see ASE, we know that we're talking about an enzyme. Okay, that's what ASE means. So each one of these ASE ending words is an enzyme that has a very specific function in this process. For example, topoisomerase right here, this big green thing, is actually unwinding the DNA. So we know that DNA typically exists in a spiraled double helix format, but this topoisomerase protein, this enzyme, is unwinding it. As the DNA is unwound, we've got this blue thing here, and it's called helicase. This is another enzyme, and it's unzipping the DNA. Helicase is breaking these hydrogen bonds that exist between the nitrogen bases of the DNA. So it's breaking the rungs of the ladder, and we can see that happening. So topoisomerase unwinds the DNA, helicase comes through and unzips the DNA. Then we've got several other things going on here as well. We've got DNA primase, which is needed to prepare the DNA to, it's needed to read the DNA to make sure that the right RNA is coming in. So this is a process that is a little bit too complicated for the purposes of this class, but it is one that we talk much more about in the AP class. And the end result is that we end up with 2 new strands of DNA, 2 new strands of DNA. Okay, so another question, and you all should try to answer this one as well. Where does DNA replication occur? Where is this all happening?
In the nucleus?
Exactly, yeah. So we know that eukaryotic cells have nuclei. And so, of course, that's where we keep the DNA. And so, obviously, that's where DNA replication occurs. In prokaryotic cells, there are no nuclei. So this is all happening in the cytoplasm. So DNA replication occurs in the same place where you find the DNA. Great. So a little DNA joke for you here. We've got the prokaryotic cell on the left, the eukaryotic cell on the right. The pro says, you tried to copy my DNA, but you did it backwards. And the eukaryotic cell says, and? Does anybody see what I did there? DNA backwards is and. Okay. And just a little DNA joke for you all. Keep you on your feet, keep you on your toes. Okay, back to the notes. Brief interlude there. Thank you, Joseph. Thank you for playing into it. So DNA replication as a process is considered semi-conservative. Semi-conservative means half of the original parent cell's DNA is saved and used in the new daughter cell's DNA. So in this case, we can see that the original parent cell's DNA is blue here. As it gets copied, half of it is kept in each strand. Half of the new strand is blue. And then half of the new strand is red. So it's coming, this is the new DNA. So this is the process of replication being semi-conservative. It's not totally conservative because in that case, we would keep the entire parent cell's DNA in one strand and make an entirely new strand of new DNA. But it's semi-conservative because each newly synthesized strand has half of the parent DNA. And I love that Alicia told us that ACE means enzyme. If you were unsure about that, you should definitely write it down. ASE. This is a common suffix indicating an enzyme. And we know that enzymes do special things. They speed up reactions. They're a catalyst. All right. So here it is again. An enzyme called helicase unzips the double helix by breaking the weak hydrogen bonds that hold the nitrogen bases together. That's the first, for the purposes of this class, that is the first step of DNA replication. Helicase. So by unzipping the double helix, what we're doing is we're making each nitrogen base available to be read, and then it can be copied. So when they're together, when both nitrogen bases are hydrogen bonded together, we can't get inside to copy it. We can't get inside to even read what it is. So we have to unzip the DNA first. The second step, another enzyme called DNA polymerase will come in and add complementary bases to the exposed strands. So we can see what we're talking about here. This C here is exposed. This T is exposed. This C is exposed. And so we need an enzyme called DNA polymerase to come in and start adding bases to this C, to this T, and to this C. We know which base to add because the bases are complementary to one another. So adenine is complementary to thymine. Guanine is complementary to cytosine. So anywhere there's a C, I need to add a G. Anywhere there's a T, I need to add an A. Anywhere there's a C, I need to add a G. And so on and so forth. Down here, I've got a G, so I need to add a C. Then I've got an A, so I need to add a T. Your gizmo is really going to help you to see how these base pairs are complementary. They fit with one another. So again, structure determines function.
Sir,
Yes.
could there be like two A's on the same strand?
You mean like two A's back to back?
Yeah.
Yes, that's fine. In fact, there are some. Um, there are a lot of strands of DNA that actually have to leave the nucleus, and they travel to other parts of the cell, and they have these long strands of nothing but A's on their tail, and it's called a poly A tail. And the reason it's there is because the longer the DNA is outside of the nucleus, the more likely that it will start to degrade, and it'll degrade from the ends. So if we put a lot of A's on the end of the DNA, A's that we really don't need, then even as those A's start to get degraded, it doesn't hurt the actual DNA. And so it's called the poly A tail, and it protects the DNA.
Can the same be said for the other types?
Yeah, you will see it. It's just, I mean, it's unlikely. You will see, you can absolutely see like several T's in a row, several C's in a row. It's less common than the poly A tail, but you can see that. Yeah, good question. Okay. Another thing to point out here, and we can actually see it in this image as well. The two new strands of DNA are not exactly the same. One of them is called the lagging strand, and one of them is called the leading strand. This is getting really far ahead. This is like pretty high level. We do talk about it in AP biology, and it's difficult to understand. But we can see here is that this leading strand is being copied in the exact same direction that the DNA is being opened up. So as this helicase is unzipping the DNA in this direction, on the leading strand, DNA polymerase just follows right behind it, and it's adding new base pairs in that exact same direction. In fact, we can see it with this arrow here. On the lagging strand, it's much more complicated because of the directionality of DNA. So what we actually have to do on the lagging strand is we still add new DNA, but what we need to do is kind of separate this DNA polymerase from the DNA strand and then put it back over here and then add DNA in this direction. So DNA can really only be added in one direction, from 3' to 5'. And so this creates somewhat of a conundrum. And that's why we end up with a lagging strand that is kind of moving backwards and a leading strand that is moving in the exact same direction of the helicase. So this is an extremely complicated process, and this process alone is something that scientists are still investigating and still learning about, because the fact that this is able to happen, and it happens constantly and it happens without error, is really a miracle. Okay. The third step, we've got yet another enzyme called ligase, which is kind of like the glue. It comes in and it reforms those hydrogen bonds that were just broken. Essentially, it comes in and it rezips the DNA. So helicase kind of unzips the DNA, and then we get ligase, which comes in and zips it back up. So, excuse me, ligase is basically the glue, comes in and rezips the DNA. It reforms hydrogen bonds between the complementary base pairs. It's beginning to look. Okay. And then the last step, essentially, another DNA polymerase. So they're both called DNA polymerase, but really one is called DNA polymerase 3, and then another is called DNA polymerase 1, but they're both called DNA polymerase. This one comes in and it proofreads the strands to make sure that there are no mistakes, aka mutations. Mistakes, that's funny. And so what you even in this example can see is how the complementary base pairs line up. We've got CG, AT, GC, CG, AT, GC, CG, AT, GC. So this final step, this proofreading is so important, because as we have seen before, mutations can be fatal not only for the cell, but for the tissue that the cell is a part of, and potentially even for the entire organism. So mutations need to be taken care of as soon as they are discovered. And the cell can take care of these mutations in a number of ways. It can choose to try to fix it by removing the mistake and replacing it, or it can just say, this mistake is too severe. This cell just needs to, it can trigger what's called apoptosis, which is basically programmed cell death. So it'll say this cell needs to go ahead and remove itself. Okay, so this is what the process looks like. Essentially, we've got a ladder, right, which is basically what DNA is. It's a ladder that's twisted into a spiral called a double helix. We cut the ladder in half, we split it up. Then we come in and we add new rungs and new backbones to the ladder. Now we should end up with 2 ladders. Each one has half of the old ladder and half of a new ladder. This is a really simplified version, but this is basically the process. How long is this?
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay. So that was a pretty comprehensive video, explained quickly. I had to put it into hyperdrive so we can get to this next part because I want you all to practice. So if we've got complementary base pairs here, we've got a parent strand of DNA and a complementary strand. I've got a T here, so what should I have in the complementary strand?
A.
A, good. Let's keep going. Let's be quick with this. What's next?
T.
Good, there should be a T. Then what?
T.
If I've got a C here, then what letter should I, or what base should I have under here?
G.
G. Then what?
G.
G. And then
C.
C. Then
T.
T, good. Another T. Another T.
A.
A.
G.
G.
C.
C.
G, T.
G, T. Good. All right. So it's very simple once you can remember how they match up, how they complement each other. A goes with T, T goes with A, C goes with G, G goes with C. So can we do this one? Now this one, okay, keep going. This one should be quick. Come on. Someone just knock out the whole thing.
A, T, G, C, T, G, C, A, T, T, C, C, A, G.
Excellent, excellent, excellent.
That's
Perfect. Now this is a fun fact. Again, we talk about this in the AP biology class, but this is a really important part of DNA. It's called the Tata box for obvious reasons. It's TA. A lot of our DNA, a lot of the genes that our body has, so the important parts of the DNA that actually code for some type of protein, they begin with this Tata box. They begin with TATA. And this signals to our cells that the next portion of DNA that comes after this is important. So we need to take care of it, and we need to make sure that it gets properly copied. So this Tata box is really, really important in our bodies, in our cells. All right. So this is the same strand that Castro just complemented for us, but I want to push you all a little bit. If I've got this DNA, AT, GCT, GC, ATT, CC, AG, how can I get that into mRNA? We do the exact same thing. However, in mRNA, we don't have T's, we have U's. So anywhere that I would put a T, I need to put a U.
Can we
So we should go right here.
U,
U, good.
UA, CG, A, CG, U, AA, GG, U, C.
Excellent. Good. So Castro caught on really, really quickly to that. We're going to continue to practice with this. And we're going to talk much more about the RNA side. But what he just recognized is that, again, RNA does not have any T's. So anywhere that I would have a T, I need to have a U instead. So instead of A being a complement of, instead of T being a complement of A, I've got U as the complement of A. Then here T goes to A goes to U, T goes to A. The C's and the G's are the same. We'll practice much more with this. It's 9:52, so you all should be getting ready for third block, and we'll talk more about this tomorrow. But good job today. If you need any help with the gizmos, please let me know. I know we kind of ran through that quickly, but if you need help accessing it, do not hesitate to reach out. We will be using them. I think they're going to be fun, something that you all enjoy. So yes, I'll end there. Have a good day.